That's good. Welcome, everyone. It is 10:01. We're happy that we're all safe and sound and slowly recovering from the stressful events last week, and hope that the next weeks are not going to be as stressful as much as we can see. But there's hope on the horizon in the sense that maybe by June next year, we'll all be vaccinated. But it will take long, so we will not be coming together soon. I spoke yesterday to the president of the ASMS, Rick Yost, who said while he would hope that the ASMS would be in person in Philadelphia, he does not see it happening. So until June, most likely, we will still sit in front of our screens and smile at as much as we can without seeing people a lot. But it is my pleasure to smile today at Chuck Burant, who's an MD PhD at the University of Michigan. Dr. Burant earned both his medical degree and his doctorate of philosophy in molecular and cellular biology from the Medical University of South Carolina in Charleston in 1987. His internship and residency were served at the best state of the United States, that is University of California, San Francisco in 1989. For no reason, he left the state and completed his fellowship in the Department of Medicine Endocrinology section at the University of Chicago in 1992 and joined the University of Michigan faculty in 1999. He is a professor of internal medicine, section of metabolism, endocrinology, and diabetes. He holds the Robert and Veronica Atkins Professor of Metabolism Endowed Chair, and his clinical interests are in the care of patients with obesity, type 2 diabetes, and related conditions. His research program integrates molecular phenotyping, that is genomics, transcriptomics, proteomics, and transcriptomics, with dietary, clinical, and biobehavioral phenotypes to understand the development of obesity, insulin resistance, and diabetes in both people and mice. He also has an interest in intermediate metabolism of beta cells and how nutrients and insulin resistance modulate insulin secretion. He directs the Michigan Metabolomics and Obesity Center, the umbrella organization for the NIH-funded Nutrition Obesity Research Center and Michigan Metabolomics Resource Corps. With that, I welcome Chuck Burant, with whom I have worked over the last 8 years in the metabolomics centers of the NIH, and I give you the floor.
Thank you, Oliver. It's really, it's nice to see everyone, or sort of see everyone. I wish I was there. Obviously, it's life would be much better. Oh, what did I do here? Screwed it up. Let me. All right. I have to move things around a little bit because it seems like a too. All right. Is this look okay on your screen?
I see you, but not your screen, not your.
Yeah.
On the share screen, there's a green button. I almost saw it, and then it went away.
Huh. So I sort of have, let me just get out of this and retry.
Okay.
Sorry.
That's all right.
Despite all my my. Okay.
There you go.
Okay, you can see it now.
I see your PowerPoint.
See my PowerPoint, and now is it full screen, or do I need to swap it now? There.
It looks like now it's a full screen.
All right, excellent. So again, thanks everybody. This is I'm Chuck Burant, and I work in this nice building over here. It's called the Burant Tower at the University of Michigan. It's a fairly new lab, and we have all our metabolomics set up there. There's clinical work goes on in the first couple of floors, so it's a very cool place to work. That's my conflicts of interest. I'm trying to get rid of all of them. I don't like them anymore. And the first thing I do is I want to thank everybody who did all the work, because I usually rush through the end of it and I never get a chance to really thank everybody who's really who gets to do all the work. A lot of this is going I'll be talking about stuff that Mary did. Actually, it's my wife who runs my lab. Katie Overmeyer, Charles Evans, Ray Young, and Johanna Fleischman has some work in here as well. And my close collaborators, Steve Britton and Lauren Koch, who actually just moved to the University of Toledo about a year ago. So I'm going to go through and I'm going to talk to you about cardiorespiratory fitness and what we've been doing to try and understand what cardiorespiratory fitness is and really why it's so good for you. So really, cardiorespiratory fitness, or VO2 max, is really the relative ability of the circulatory system and your muscles and your lungs and stuff to get oxygen into your heart, your skeletal muscle, your lungs, all those things that you need to run hard. And the essence of it is really to be able to transfer electrons from carbon to oxygen and reduce the oxygen. That's really the basis of life, right? It's all these energy transfers, and everything else derives from that. So usually you determine cardiorespiratory fitness by running people on treadmills of progressively harder and harder exercise regimen, and you can measure how much oxygen is being taken up and how much CO2 is being disposed. And so we have a nice lab. We actually have a couple labs now that we do this. This is Heidi and Chrissy, who are really integral to doing the clinical work. I'll show you some clinical work as well as some of the animal work we're doing. But we really want to know why this is good, because cardiorespiratory fitness is actually your best estimate of how long you're going to live. Actually, more than anything else, how long your parents lived and things like that. Measuring your oxidative capacity or cardiorespiratory fitness is a great measure. So if you have high metabolic equivalence, is just a way of designating what your cardiorespiratory fitness is. If you have a high one versus a low oxidative capacity or fitness, you can see that your relative risk of death, age-adjusted risk of death, is markedly lower. Now, this is a Gaussian distribution of METs, so most of us live here. But not only do you live longer, but it's really related to your cardiometabolic health. Now, the first question I always get about this is you have a certain amount of what your VO2 max is. What does exercise do? What is on top of this? Because what I'm going to try and convince you is that it's really much more important to be born right as far as your cardiometabolic health than it is to exercise. And so this is a study, and there's a number of studies like this, that if you have high intrinsic respiratory fitness and then you do physical activity, your mortality hazard ratio is not changed. So if you have a high intrinsic oxidative capacity, physical activity really doesn't help you, even though if the highest ones. Now, if you have medial fitness and you're in the middle tertile, you do get some effect, but it's only if you're really, really highly active. And if you have low fitness, these are the people who actually get the most benefit out of exercise. Excuse me. So, a lot of these set recommendations from the government about how much exercise you should do, 30 minutes, a couple times a week of moderate physical activity, it does help, but it helps mostly those people with low intrinsic oxidative capacity. And I think there's lots of data that would suggest that. And in fact, the amount of exercise that you do is probably, the recommendations are too much. Likely you can get away with a lot less activity and improve your metabolic health if you have low cardiorespiratory fitness at baseline. I'm happy to talk about more of this when I get through this. All right. So I told you cardiorespiratory fitness, your risk for cardiovascular events goes down if you have high cardiorespiratory fitness. Your risk for diabetes, I'm a diabetologist, I like showing this, that if your metabolic equivalents are at the lower end, you have a high risk of diabetes. And as your cardiorespiratory fitness goes up, you get a marked reduction in the relative incidence of type 2 diabetes. And this interacts with age, but sex, race, and things like that, the relative efficacy is there no matter what your age, your race, and things like that. So it really is across the world. A lot of these kinds of studies have been done and shows that how hard you can run on a treadmill is predictive of your cardiometabolic risk. And it's actually I like this slide when I came across it. It looks like even if you have a low versus high BMI, if you have a higher oxidative capacity or fitness, your cost to the healthcare system goes down. And you can see that even obese individuals with high oxidative capacity actually cost a lot less money. And in fact, these individuals are these more healthy, obese individuals. So it's good to have a high cardiorespiratory fitness. All right. Now, there's been a number of studies, not a lot of great studies, but a lot of small studies that if you compile them together, shows that cardiorespiratory fitness has a high genetic component. It's usually twin studies or family studies. Klobuchar has done one of the larger studies. And if you put all these studies together, meta-analysis is about 50 to 75%. Most people put it nearer 75% of your cardiorespiratory fitness is genetically determined. So most individuals, on average, a person can increase their VO2 max or fitness by about 8% is the average. Now, people with very low cardiorespiratory fitness, intrinsic, they can have a much greater increase. They can almost double their VO2 max. But if you're, again, in my estimation, if you're in the upper half or maybe even the upper 2/3, you get very little health effects from exercise. Now, I know that's heretical to a lot of people that's watching right now, but the data would support that statement. Okay, so what we want to know is, okay, so what is intrinsic cardiorespiratory fitness and why is that so good for you? All right. Well, as I told you, it's really the ability to transfer electrons, right, from oxygen to turn to carbon dioxide and water. And we know that a number of disease states that we know really are reflected in the changes in the intrinsic capacity of the mitochondria to oxidize. And really it's fatty acid oxidation. I'll show you some of this, but it really looks like your capacity to oxidize fatty acids really correlates with your intrinsic cardiorespiratory fitness. And so if you have inflexibility and substrate switching, when you go from fasted to fat and you can't change your fuel utilization from carbohydrates to fat, you tend to have less metabolic health, you're more insulin resistant. We all know that there's certain signatures in the blood that are associated with the relative risk of type 2 diabetes. These are usually fatty acids or amino acids. Branched chain amino acids are one of the things that a lot of people look at. High branched chain amino acids in your blood is associated with an increased risk of diabetes. And I'm going to show you some data later that's actually is really one of the best markers of having a low cardiorespiratory fitness. And then changes in the expression of oxidative genes in the skeletal muscle, you can show that there's lower ones. So I'll show you a little bit more of this data. Okay, so the way I got into this was I saw Steve Britton, who was at the University of Toledo, came and gave a talk at the University of Michigan. And I've been working on skeletal muscle metabolism for my whole career. And he gave this, and I've known about cardiorespiratory fitness, but I didn't know about his animal model. What Steve and Lauren did was got a number of rats from the NIH, a genetically heterogeneous founder population. It's called the NIH rat. And what they did is they ran about 188 of them on treadmills, and then they found the 13 best males and 13 best females, and the 13 worst males and females as far as running on a treadmill. And what they did is they ran them on a treadmill where they had a 15-degree incline, and every minute they would make it go faster and faster and faster. And in the beginning, they did it 5 times, and now they do it 3 times. And the best run is the rat's oxidative capacity. And they've done that the distance really does directly correlate with the fitness. And so what they did is they took 13 of these highs and lows, and they started breeding them together in a rotational breeding scheme to try and keep the inbreeding coefficient low. Over the time, what happened is that they had a selection. There's a technical thing. This is actually an operator condition. People who were running the rats here didn't like to push them so hard, so they sushed them along instead of making them run a little bit harder. But you can see that over these 35 generations, there was a marked increase in the amount of running capacity in the HCR line, the high capacity running line, and there was a decrease in the low capacity running. These guys now, we're actually out to generation about 42, I think. These guys can run about 12 times farther, and their actual work that they do is about 8 times more. Now here is a bunch of inbred strains of rats, and these are the running capacity. And so you can see that while this is statistically going down, there's some technical aspects to it. If you're a low capacity runner, some won't run on the treadmill at all. About 15% won't run these days. Here, only a couple percent won't run. And so if you don't run, you don't get to be in the breeding scheme. We don't tell them that before. But you can see is really we're selecting for a high running capacity, much more than we're selecting for low running capacity. Okay. So the HCRs, the high capacity runners, really do phenocopy the things that we know about in healthy humans. So they have less visceral adiposity, their lipids are down, the insulin levels are down, glucose levels are lower in the high capacity runners versus the low capacity runners. And their whole body O2 extraction, the amount of oxygen that they can utilize is higher, their lung diffusing capacity is higher, and things like that. And the cool thing is that they do recapitulate the increase in natural lifespan that we see in humans with high oxidative capacity. So here's one of 2 studies that were done in different places in the world, got basically the same results. And I'll show you this one. This is the Kaplan-Meier death curve. And you can see that the median lifespan of the low capacity animals is 24 months, where the high capacity animals, it's 34 months in this experiment. So anywhere from 25 to 40% increase in survivability. Now what's really cool, and this is actually a really, really important point, is that if you run a group of rats from any sort of generation, and you run them on a treadmill, you run them at 6 weeks of age, you run them at 6 months of age, you run them at a couple of years of age, the rank order of running tends to stay the same. These are genetically heterogeneous, but the intrinsic capacity of that rat throughout its lifespan relative to the other rats tends to stay the same. And so what you can see here is the longevity, the lifespan of months, and let's duplicate here, that the low capacity, the lowest of the low capacity animal lives shorter amount of time than the highest of the low capacity line. And the lowest of the highs live shorter than the highest of the highs. And this linear relationship is really kind of interesting. So what you're really doing, if you think about it, you're selecting for longevity. Simply by running an animal 3 times on a treadmill, which I think is just fascinating. And so you can see that, you know, the rank order stays here at different months of age. They ran them each time. What you can see is that by 34 months of age, only a subset of the high capacity runners are still around. Now I'm going to show you later that the gene expression profiles of the high and low lines are different, but the gene expression profile along the running capacity in the high line or the low line actually parallels the running capacity. So the genes that are upregulated in a sort of a rheostatic mechanism. And that's what we're really trying to figure out. So whatever's going on, the genes that cause this high running capacity are differentially regulated across the running capacity. All right, so as I told you earlier, in humans, it's known that people who have a higher oxidative capacity or cardiorespiratory fitness, VO2 max, same word, they have a higher capacity for fatty acid oxidation. So Katie Overmeyer, who was a graduate student in the lab, actually is now running Josh Kuhn's Metabolomics core at the University of Wisconsin, and Charles Evans, who's a close colleague, we've been working together now for about 15 years, who's the true mass spec guy, did experiment with Nathan Shee in our animal core, and just we can actually, there's a way of deriving how much. And so you see the high capacity runner burns carbohydrates, carbohydrate utilization goes up and they start running, and they continue running. The low capacity runner uses more carbohydrates and then they peter out. And the low capacity runner, relative to the high, cannot use as much fatty acids. And so this high capacity runner here can use a lot, these guys can use a lot of fatty acids. And what they essentially do is spare glycogen. So once you start hitting your lactate threshold, that's really when you're going to stop running, because you're now likely using glucose from glycogen to supply or to make ATPs from glycolysis. That's really what the lactate threshold is you really reach your capacity to generate ATP in the mitochondria, and you have to start using the glycolysis in the ATP that's generated there. And once you use all your glycogen, then you can't make any more ATP, and that's when you stop running. Okay, so really, so at 10 minutes of running, the LCRs stop running pretty much, and they've depleted their glycogen, where the high capacity runners do not deplete their glycogen until they really get to to a high enough level where they can't generate enough ATP in the mitochondria. All right, so here's the genes. So, or the gene pathways that are upregulated in the skeletal muscle of the high capacity runners versus the low capacity runners. So the ability to do fatty acid oxidation, branched chain amino acid oxidation, TCA cycle, there's some peroxisomal utilization of fatty acids that are probably a little bit different as well. But these are the pathways that are upregulated consistently in the high capacity runner and the low capacity runner. And these pathways also correlate with the running capacity in the high line, within the high line and the low line. So those genes that are differentially expressed actually then correlate also with the running capacity. And this is not the first time that these pathways have shown up. This is from Mary Elizabeth Patty's lab a couple of years ago, looking at people with insulin sensitivity and insulin resistance. And the top pathways in the gene expression profile are fatty acid metabolism, malate or TCA cycle metabolism, branched chain amino acids, and propionate metabolism, which is still intimately involved with or related to the branched chain amino acid metabolism. So that actually makes us feel good. And then, as I told you earlier, that blood metabolites associated with diabetes risk are branched chain amino acids, aromatic amino acids, different kinds of lipids, and things like that. And when we look at the metabolites that are different in the blood of the high capacity and low capacity animals, it really is aromatic branched chain amino acids and fatty acids. So I think that that the animal model we have really recapitulates what has been described in humans. So we've been really excited about that. All right, now quickly I'm going to tell you this is a paper that's published. You can go back to it, look at the details. But one of the things that we did with a grad student in June Lee's lab, who's a collaborator here in human genetics, is that we took the high capacity and low capacity animals and did young animals. They were, I think they're about 8 months of age, and then old animals at about 22 to 24 months of age. And we looked at them at at baseline, at old and young age. And then we looked at them after acute exercise and looked at the differences in gene expression across these things. And what you can see is that the lines diverge with age. So here's the HCR and the LCRs down here. Here's LCR, here's HCR. Then as they age, you can see that they diverge, okay? And then if you look at the aging, it only has a small effect on acute exercise, the genes that change in acute exercise. Now the genes change, but the magnitude of the change is not different. So when you exercise these guys acutely, the same genes go up with exercise in the highs and lows, but the magnitude of the change is different, depending upon your age and how much you can run. So we think that when we look at the gene sets that are different between the highs and lows, there's a certain set of genes that I showed you before, but the genes that change when you do acute exercise, and indeed when you exercise train, are the same in the highs and lows. Those are not affected, and they are different genes. The genes that change with training are different genes than are associated with the differences between the lines. So I think that's important because that may tell you why exercise is not that effective in people with high oxidative capacity, because they already have a lot of good mitochondria, at least that's the way we look at it, and that you actually just make more of the kind of mitochondria that you have when you exercise train. You don't change the intrinsic quality of the mitochondria, you just make more of what you have. And that's what exercise training does. All right. So these guys live longer. So one of the things that we want to see is, can we see the reflection of the aging effect in the transcriptome? So what we did is we looked at the genes. So here's the highs and lows, acute exercise, not exercise. And once you adjust for all these kind of things, we could actually identify age, genes that change both in the high capacity animal and the low capacity animal. And then we could look at the relative change at aging. So a lot of genes in skeletal muscle decrease as you age. But if you look at the decrease in the LCRs versus the HCRs, the LCRs have a greater slope. And this slope suggests to us that this is showing that there's an accelerated aging in the low capacity runners versus the high capacity runners, or vice versa, the high capacity runners are protected from this age-related gene change. And if you take it just from the slope, and we've done this with a couple of different data sets now, if you just look at the slope, it's 26% greater aging in the LCRs, which actually matches up pretty good. What we see is the differences in the gene expression profile, or excuse me, a difference in the age. So gene expression profile slope matches up pretty much with the change in the longevity of the HCRs compared to the LCRs. All right, so quickly, because this goes back to some other things I want to get back to, but one thing that causes a increase in longevity in most species, mammals included, is caloric restriction. So we decided to see what happens if we do caloric restriction in the high capacity versus the low capacity animals. So what we did is a 40% caloric restriction in the high capacity and the low capacity line in 8 weeks, and we did it for a year. And so we did 12 animals in each group. And so here's the low capacity animals. They have a higher body weight than the high capacity animals on an ad-lib diet. So that's here's low and high ad-lib. And you can see that the caloric restriction in the low capacity runner reduced its body weight, and the high capacity runner reduced its body weight. And they were a little bit different, but they're pretty much the same, the amount of weight loss. This brings up another really important point, is that the high capacity runner seems to waste energy, okay? So we have really good data that the high capacity and low capacity animals tend to eat the same amount of food. However, the high capacity runner gains less weight than the low capacity runner. Now we've done bomb calorimetry on their feces, and there's no change in the absorption of food. It just seems that the easiest way to say it, there's some sort of wasting of energy in the high capacity animals versus the low capacity animals. Now, I think this is important to think about is the relative inefficiency. So these guys they have the capacity to burn fatty acids better in their mitochondria, but they're less efficient at it because, you know, if you just think about it, they're eating more, but they gain less weight. And there's something going on in there. And this is one of the really intriguing things. So you can see the body weight changes and stuff like this. One of the things that we do see consistently is the high capacity runners or low capacity runners have a slightly larger amount of lean mass compared to, and individual muscles tend to weigh more even when you normalize it to the total body weight. All right. So if we do a PCA plot of the age-related genes, or excuse me, actually, this is all the genes. Skeletal muscle transcriptomics, 40% caloric restriction. Here's the LCR's ad lib, here's the HCR's ad lib. And what you can see is that when you do caloric restriction, you get in the PC3 space, you get a change in the gene expression that goes up. The high capacity runners don't change that much. So the centroid between these PCR clusters actually changes much more. It's almost like the HCRs are already pre-calorically restricted. Now, I told you before that there's an inefficiency in the metabolism, it seems. And so is this inefficiency in metabolism being reflected in the gene expression profile? Somehow the nucleus is actually sensing a change in the energy intake as if there's not enough energy. So I just want to leave that thought with you, trying to figure out exactly how are we getting from, you know, from the intrinsic breeding into the changes in gene expression. All right. Same thing will happen in the plasma metabolome. We have the lipidome. We've done this in skeletal muscle as well. And again, the low capacity runners versus the high capacity runners with caloric restriction. This is in this case is in the PC1 space. That the high capacity runners tend to have a shorter distance than the low capacity runners. Again, supporting the notion that there's a difference in metabolism, and that the high capacity runners and the low capacity runners actually get a little bit closer together with caloric restriction. All right, so these are rats. I'm going to come back to the rats, but I'm just a short diversion to ask the question, well, does this happen in humans? So with Ray Jung, the head of our geriatric center, and with Heidi Igle-Rieger, who's in my lab, we created a study with the hypothesis that high VO2 max in humans will modulate age-dependent changes in clinical endpoints and metabolic endpoints. And so we took 100 healthy middle-aged subjects, aged 45 to 60, and then Ray came up with or derived a multi-system assessment tool for aging. So this contains like HDL levels, blood pressure, heart rate, all those things that we know change with age, and developed a score to rate everybody who came to our study, gave them a score for all these different parameters. And then what we did is we did plasma metabolomics on them along with this MSAT, and then we ran everybody on treadmills and got their oxidative capacity, did multivariate analysis, and asked the question. So I won't go through all the studies, the data, but let me just show you a table of the end result. So here are all the variables that changed significantly with either age or VO2 max, either or. And then what we ask, is there an interaction between age and VO2 max? So we know that, you know, age, you can't change your age, but you can have a different VO2 max. So if you look at all these things, and this includes a number of metabolites, fatty acids, some aromatic amino acids in here, and then these clinical measures, your grip strength, your serum creatinine levels, how many wrinkles you have, your creatinine clearance, which goes down with aging. And you can see that here's these changes. These go down with age. These couple of things go up with age. And then here's the change if you have a higher VO2 max. These things tend to be lower with a higher VO2 max as well. But if you look at the interaction, what this interaction term means is that how does having a higher VO2 max change the age-related changes in these different parameters? And all these are very significant. I plopped a couple in here just because I wanted to see, it. But you can see that for the most part, there's a significant interaction. And this term, the beta value, is positive compared to the negative beta value with age, suggesting that the higher the VO2 max you have actually slows the age-related change in these clinical parameters and these metabolite parameters. Okay. Chinitha Thanusan, who is a MD PhD student in the lab, did another study. We have a large weight loss clinic that we run, and we took lean and obese individuals, and we phenotyped the heck out of them. And you know, so obviously the obese people, we did a baseline, and then after caloric restriction, just like we did in the rats, we wanted to do it in humans. So we put them on a very low calorie diet, and they lost on average about 17% of their body weight in about 4 months. And we ran them on treadmills at baseline, we ran them on treadmills afterwards, we drew their blood, we did muscle biopsies, and I'll show you a little bit of that data. So first thing to know is, a lot of people ask this, so I always point it out, is that your VO2 max, when you're an obese person and you lose 17, 18% of your body weight, you do not change your ability to run on a treadmill. So this is, again, this goes to this genetic thing. It also goes to the question, are people who have, we know that people who are obese have a lower intrinsic oxidative capacity. The question is that low intrinsic oxidative capacity because they're obese, or are they obese because they have a low intrinsic oxidative capacity? Now, actually, I favor the latter. And I think, the data like this, but it's really hard to do because, they co-develop. So obesity and VO2 max. So we're doing some pediatric studies right now to see if we can actually tease out some of these changes in kids as young as 8 years old. Okay, so, but when we do this, and we do plasma metabolomics again, and this was a targeted assay, and we looked at the acylcarnitines. Remember before, acylcarnitines were one of the things that changed with age. And so here, age causes an increase in in these people. Age is associated with an increase in these acylcarnitine levels. And VO2 max in this case is related to a decrease. So this is a MANOVA value. So we actually compiled all these metabolites, used them as a unitary value, did the MANOVA, and there's a significant change in intermediates of fatty acid metabolism with both age and VO2 max. And then if you do the interaction term, it's really highly correlated. This is before weight loss. And so this really tells you that that again, as in the other experiment, that higher oxidative capacity can modulate age-dependent changes. Now, if you do it after weight loss, what we see is the same thing. This is, we see the same age dependence even after the obese individuals lose weight. VO2 max now didn't have as much of an effect. There's smaller numbers here. You can see that the age-dependent change is reversed, that the interaction term tends to be positive, but did not reach statistical significance in this. We're not quite sure if it's our small numbers. We fully expected to do it, although there's some funny things about when you lose weight and you change your metabolite levels, that sometimes it can actually be working against you. But our thought is this, is that if you look at metabolism and age and you have a low cardiorespiratory fitness, your age-dependent change in metabolism goes down. If you have higher cardiorespiratory fitness, like in the high capacity animals, rats, you have a modulation. The slope of this line is attenuated. Now, if you're an obese person and you do caloric restriction, what we believe is your metabolism gets a little bit better, but it doesn't seem to change your slope. So caloric restriction can make you younger metabolically, but it's not going to change the slope. So this is what caloric restriction tends to do. We believe this is what's happening in the low capacity animals. You caloric restrict them, their metabolism, their metabolites, their gene expression profile tends to get closer to the HCRs. And so if you happen to have a low intrinsic cardiorespiratory fitness, if you want to make yourself live a little longer, you have to calorically restrict. At least that's my suggestion to my patients, at least. All right, what's the, how do we get to this? All right, so we have this animal model, right? And so the cool thing about this animal model is that we have running phenotype on about 14,000 animals. And we have DNA in about 3,000 animals. So every rat that was bred in this experiment, we have DNA for. And so we can make some insights into the genes that are associated with it. And I'll show you that now. All right, so what we did, and we actually had to go to Czechoslovakia and have somebody open up their freezer because the NIH rats are derived from a random crossing of 8 inbred strains of rats at the NIH. So these 8 inbred strains of rats were just crossed. Now, unfortunately, 4 of the strains of rats that were around in 1989 when this was started, or excuse me, 1996 when this was started, no longer exist. And so we wanted to do whole genome sequencing. And we actually found our last sample. Somebody had an old liver in a freezer in Czechoslovakia. And we were able to do whole genome sequencing. And so, and we found about 650,000, sorry about that, 650,000 SNPs across the genome in all 8 of these inbred strains. So now we know the genetic variation that went into making these animals. So essentially what we could do is do a selection. We're doing the selection, and we can paint the chromosomes to be able to see which parts of the chromosomes are associated with the running capacity. So we could do this for a quantitative trait, and then we also did it as an EQTL. So we had 4 undergraduates for 1 summer. We bred some animals, and they ran 645 animals on treadmills 3 times over the summer to get their best running capacity. And then we crossed, so this is the running capacity in the F0 and the founders. The F1 males and females, you can see the running capacity is nowhere near the highs. And then we got some in the F2 that were getting a little bit higher, but you see there's a nice range of running capacity now in these F2s. And if you look at the heritability of distance, it's at minimum, we think it's much more, but it's just the numbers. A minimum of 37% of the running capacity in these animals are genetically driven. The heart rate seems to be higher. The percent fat is highly heritable, correlates with the running capacity. But interesting, the body weight doesn't correlate very well. And this is probably because there's a lot of noise because as the fat goes up, the lean mass tends to go down. And so then the body weight is an average of both of those things, essentially. So that's why we think the body weight is not as heritable in these animals. If you look at the distribution of the running capacity and you use some old equations, we think that there's 4 to 8 main effect genes that are upstream that are associated with the phenotype of running capacity in these animals. All right. Now we have 600 some animals, 612 animals that we were successful in getting RNA out of their skeletal muscle. And if we look at the across running capacity, which gene and gene pathways are enriched across that running capacity, it's the same ones: branched chain amino acid oxidation, mitochondrial oxidation, fatty acid oxidation, all these different things that we see before. And just like the correlation, it's exactly the correlation along each of the lines themselves. When you actually combine these lines, you get the same thing. It's actually not that surprising, but it was very, very satisfying to see this. So we wanted to know these, and I apologize, I put it in the wrong place. Oh, I forgot a slide. Anyways, so we also looked at the correlation between the running capacity and the metabolite levels within the skeletal muscle itself. So we did both targeted and untargeted metabolomic profiling on these guys. I'll just show you the results from the targeted stuff. But if you look at the, if you take, so we did is took a specific metabolite, asked which genes correlated with the levels of that metabolite, and then asked what pathways are enriched in when you select a gene solely by the correlation with metabolite levels. And what you can see is that valine, leucine, isoleucine degradation is associated with ATP levels. FAD, which is actually the #1 differentially expressed metabolite between the high and low capacity animals when you just look at them straight. Branched chain amino acids are in there, methionine and panthenoate. And this actually comes out, it's a very interesting metabolite as well. Fatty acid degradation, similar metabolites, some of the metabolites that are inversely correlated, you can see here. Propanoid metabolism, oxidative phosphorylation, spliceosome. This is actually, we always get a signal for splicing, and it's actually really interesting, so I'm going to come back to that. But this seems to be mostly positively regulated, with those splicing is positively associated with those genes that are negatively associated with these other things. So 2-PG, glutamine malate, those things tend to be negatively associated with these main pathways that we usually see upregulated in the HCRs. So now we have some idea that the levels of metabolites in the skeletal muscle seems to be related to the levels of the gene expression profiles, or are the gene expression profiles actually related to the levels of metabolites? So, all right. So with Shweta Ramdas, who's now a postdoc at, I think she's at she at Davis? No, she's at San Diego. She's getting a PhD in human genetics and working with my friend Jun Lee. And by combining the quantitative traits of running capacity and the EQTL with the transcriptomics, we could find 3 high-quality gene expressions or high-quality loci in the genome that are associated with running capacity. One is ACAT-SB. It's a gene we've been interested in for a long time. It's increased in expression in the best running animals. There's a nice allelic variant. I'm going to come back to this. A second gene is mitochondrial ribosomal protein S10. It's actually decreased 50% in all the high running HCR animals. Seems like a very early selected gene that is associated with, that decreases are associated with better running. And then we have this long non-coding RNA that we really don't have much idea about. So what's really kind of cool about the about ACAT-SB is if you take a look at it on Western blot from isolated mitochondria, what we see is the HCRs have a band that's the same as in the LCRs. But there's actually a heavy splice variant of ACAT-SB in the high capacity runners and low capacity runners. Now remember I showed you a couple slides ago that spliceosomes were one of the interesting hits that were correlated with the with metabolite levels. We're still working on this, but it's kind of interesting. But you can see that the high capacity runners tend to have 50% of their protein density is the splice variant, whereas LCRs have a much lower thing, lower amount. We set up an assay to directly evaluate ACAT-SB activity. ACAT-SB is part of the branched chain amino acid leucine and isoleucine and valine metabolism. This is isoleucine metabolism. 2-butyl-CoA is turned into tiglio-CoA by this ACAT-SB enzyme. So if you look normalized peak area when we shoot in the mass spec, you can see that the tiglio-CoA and then propionyl carnitine is actually elevated. There doesn't seem to be a change in the Km of the enzyme. It seems just to be the amount of the enzyme itself. It may have a small increase in Vmax. We just haven't done enough experiments. So we have a gene that's associated with running capacity that is in a perfect place to be an important controller. ACADSB is acyl-CoA dehydrogenase short branched. So it takes care of both branched chain amino acids, but also branched chain fatty acids. But also its substrate can be shorter chain fatty acids as well. So if you think about what we've been talking about all along, this increase in fatty acid oxidation, but also these signals that you get about branched chain amino acid levels being higher in the blood of more insulin-resistant people, but also in the low-capacity runners, it may be that this is really at the crux of fatty acid and branched chain amino acid oxidation, and that the change in this gene may be important. I'm going to tell you something. If we overexpress ACADSB by adenovirus and skeletal muscle, we turn on all the genes that we see turned on in the high capacity versus low capacity animals. So I'm just telling you, I won't show you all the data, but we're really excited about that result. So it seems like this is a really, really important molecule. Now, how this relates to the splice variant is what we're really interested in doing. So we're really trying to overexpress the higher molecular weight species and the lower molecular weight species to see if they have some differences, because it's formally possible that the splice variant may have different substrate specificities or affinities and things like that, and that could change things. But I want you to remember propionyl-CoA and propionylcarnitine. So we think ACADSB increased protein expression increases flux of this pathway. If we inject, in this case, leucine into the HCR and LCR animals, what we can see is that we get an increase in labeling of C4 carnitines and C3 carnitines, which are the sort of the pop-off valve from that pathway. And so we can actually show in vivo in the high capacity animals versus low capacity animals an increase in flux through this pathway in the intact animal. So sort of recapitulating what we see in vitro. All right. So now we have this really interesting mitochondrial phenomenon that's associated with gene expression and longevity. So a number of years ago, I had an undergraduate working in the lab, and he was interested in bioinformatics, and I sent him to the library and asked him, I want you to look up these pathways I want you to look up other data sets that are associated with gene expression that looks like the HCRs and the LCRs. And he came across a couple, and there's some really interesting ones. But this was, he came back and said, listen, there's this one that actually matches up perfectly. If you feed C. elegans, these worms, with a B12 deficient diet, they live longer and they turn on these sets of pathways. So by being B12 deficient, you can upregulate the exact same pathways that we upregulate in the HCR versus the LCRs. And these guys live longer. So I went, that's pretty cool. Further, what this is, Nathan Walholt at UMass. What they did is they said B12. B12 is a mitochondrial cofactor that regulates two ends of metabolism in mitochondria. One, B12 levels being low here, you would inhibit the flux of amino acids into this succinyl-CoA into the TCA cycle. So low B12 will increase potentially these metabolites. And low B12 here will also change tetrahydrofolate and things like that, and also change some of the metabolism of other amino acids in this pathway. And so what they did, and I thought this was really a clever experiment, they took worms and they made a reporter gene that drove one of the genes that is upregulated, one of the fatty acid oxidizing genes in the worm, They hooked up the promoter of that gene to GFP, and then they could look at the relative expression when they actually just threw these metabolites right on the worm. Said, what happens? And what you can see is this is the diet low in B12. Excuse me. This is the and if you put on propionic acid, alpha ketobutyric acid, here part of the 3D metabolism, valine, isoleucine, but not leucine, what you do is you can turn on the expression of this reporter gene. Excuse me. So it looks like if you change a flux into a B12 deficient animal, you give it propionic acid, it causes an upregulation of all these genes. So we thought that was pretty cool because ACAT-SB, which is markedly upregulated in the HCR versus the LCR, sits right here and actually is one metabolite step away from making propionic acid. So what we think is that instead of inhibiting and accumulating propionic, I'm sorry?
I have one.
So ACAT-SB, we believe, is actually causing an increase in flux. We've shown this in vivo into this pathway into here. And really, we believe that propionic acid may be an important metabolite that's associated with it. So instead of blocking here and increasing propionic acid here, or decreasing flux from above here through cystithione and alpha-ketobutyrate. Now, alpha-ketobutyrate can also turn on these genes, but this is a direct precursor to propionic acid as well. Now, I'd encourage you to go back. There's a lot more to this story. What this group believes that this propionic acid is a toxic metabolite, and that in order to detoxify propionic acid, it's indicative of having a bad mitochondria. And so there's a signal somehow associated with propionic acid that can actually increase gene transcription to, quote, repair the mitochondria. And we actually think this is a great idea. It's actually what we've been thinking about for a while. And when we found this paper, it was really exciting to us, that we believe that somehow metabolites that are coming out of this pathway is somehow going to the nucleus and signaling. Now, I've told you that we do have evidence now that if we just overexpress ACAT-SB in a normal rat by injecting skeletal muscle with a construct, we can turn on essentially the same gene pathways. Now, I want to end this talk, I'm almost done, about metformin. Now, metformin is, I use metformin as like candy in my clinic. It's a very safe drug. It's a very effective drug. And there's actually accumulating evidence that metformin can cause an increase in longevity in humans that take it. In fact, there's evidence in animal models that metformin treatment itself can cause an increase in lifespan. Now, one of the side effects of metformin is very well described, is it actually decreases B12 levels in humans. There's actually epidemiological evidence that high B12 levels in people's bloods are associated with a reduced longevity. So could it be that metformin treatment that decreases B12 levels in humans may be subtly changing these pathways and perhaps changing metabolism? That's a hypothesis. Now, there's a TAME study is being designed. I think it's actually been executed now, to see if in humans over age of 60, if you give them metformin, will you increase their longevity? Okay, so I think it's a really cool experiment, except I don't think it's going to work, because one of the things they're doing with these older individuals is they're giving them B12 replacement because they don't want to get pernicious anemia. And so I'm afraid that this may not work. All right, so how does propionate do this? One of the things is it's an HDAC inhibitor. It's not as good as butyrate, but propionate does have HDAC inhibition. So this may be a way to change the nuclear expression of genes, and HDAC inhibitors actually turn on skeletal muscle. And there's some evidence now in the literature that if you instill propionic acid into the rectum of mice, they run better. So here's our working hypothesis. We have a number of genes A, B, and C. Let's say there's 5 genes. A catus B is one of them. Mitochondrial ribosome protein S10 is fascinating. We believe this is really related to longevity. And if we have time later, I'm happy to talk about that. We think that this is signaling that these mitochondria are defective. And we've done this experiment now with Charles Evans. We fed animals deuterated water, which gets incorporated essentially into alanine. And then we took the mitochondria, ground them up, hydrolyzed and measured deuterated alanine. And what we can see is that the labeling in the HCR mitochondria is much more than the LCR mitochondria, about a 50% increase in labeling rate, which means that they're turning over more quickly. So we think that there's an increase in turnover of the mitochondria in the HCR animals. We think that the gene expression differences may be due to the HDAC activity of propionic acid to open up the chromosome and let a specific subset of genes be transcribed. And we think this actually is related to the increase in running capacity and may explain why the running capacity and longevity go along hand in hand. So I think I've said everything there. I'll stop. I know I'm probably going over a little bit. One of the things we're doing now, I'll just tell you, is that we have some experiments and we have some early results from this. We're doing ATAC-seq on the animals, the high and low capacity animals, to actually look at the chromatin state, both at rest and running. And we're actually doing some propionic acid feeding as well. I'll stop there. Happy to take any questions.
Thank you so much, Chuck. That was data-rich, information dense, inspiring, and you know, obviously there could be a lot of questions now. For everyone, Chuck Burant was gracious enough to say he would stay around for like half an hour to answer questions, so we don't have to be shy on asking questions, and he would enjoy that, he said beforehand. So there are a number of questions here. The first one from Zach Rabo: does the rate of carbohydrate uptake during exercise differ between the high and low capacity groups?
In exercise, it's during exercise, the rate of uptake. We don't, what I can say is that we don't think that there's a limitation of transport of carbohydrates into the high capacity or low capacity muscles per se. We actually think that because, you know, as you start running, there starts to be competition, as it were, between fatty acids and carbohydrates as a fuel source. And so we don't think that carbohydrate metabolism is limiting in the HCRs. It's just better it's better in the HCRs. Because if you look at the beginning of the, I don't know if I'm still sharing the screen or not, but if you look at the when we did the looking at the carbohydrate and fatty acid oxidation, it's just the fatty acids are just starting to be used better in the HCR early on in the exercise. So when we do clamps, it's actually, when we do hyperinsulinemia euglycemic clamps in these animals, it's really interesting. The LCRs look, quote, more insulin sensitive because they can take up more carbohydrate. But under these clamp conditions, we never infuse fatty acids to see how that might interact.
All right, thank you. The next question comes from Sarah Greenfield. What specific factors determine a CR fitness beyond being a rat that has been selected for certain levels of fitness, for example, in other organisms? Is it just how well an individual can run? NVMD, I don't know what that means. Just measure VO2 max?
Yeah, so the data on longevity and health is really all done on VO2 max measurements. So obviously, you know, if you have a club foot, you're not gonna be able to be measured at high VO2 max, although you can do it on a exercise bicycle. So the you know, the thought is that there's multiple factors that are associated with this, right? It's cardiac function. What's your cardiac output? How much blood can you push through? What's the oxygen carrying capacity in your blood? You know, things like that. So, and a lot of these are genetically determined as well. You know, we've been focusing on skeletal muscle. The gene expression profile in heart is similar to what we see in skeletal muscle. But if you actually take liver, you don't see any of this. You don't see, liver does not have an upregulation of fatty acid oxidation, or at least genes and things like that. So I'm not sure I'm answering the question that you wanted answered.
Well, we are part of the Longevity Consortium, and Sarah is part of that. And we have weekly meetings discussing discussing metformin, and it's complex, and we all.
It is. Well, so the interesting, one other thing about metformin, I'm glad you brought it up, is that, you know, as you know, there's some indication that metformin may mess with complex 1 in the mitochondria. And there's really interesting data that complex 1, if you mess up complex 1 a little bit, or complex 3, you actually can get longevity, even though there's some alterations in fuel utilization, and likely part of it's due to increase in mitochondrial turnover. But there's no instance in which people have shown that subtle mutations in complex 2, really, you know, the succinate dehydrogenase, that there's any manipulation you can do with that enzyme and cause an increase in longevity, which I think is actually kind of cool, because if you remember, the first dehydrogenation step in fatty acid oxidation and in branched chain oxidation uses FAD, and FAD transfers those electrons. Now that's there's a 13% decrease in the efficiency of fatty acid oxidation, because you have a decrease in, you know, you only get 1 electron being transferred instead of 2. And so fatty acid oxidation is per gram of O2 is 13% less efficient. Now, is this part of this whole, you know, inefficiency, blah, blah, blah? I also am intrigued always that FAD is the #1 metabolite that's different in the skeletal muscle between high capacity and low capacity animals. So FAD tends to be, you know, about 30% higher in the high capacity versus the low capacity.
All right. Well, Tong Shen had a question about the vitamin B12. So she said, well, you know, animals and perhaps people who live longer and have this pathway alterations might have less vitamin B12, but, you know, shouldn't that also be harmful? And that's the reason why then there was a replacement in addition to the metformin in the human cohort.
Yeah.
So what are the, at which levels would vitamin B12 deficiency become harmful? Yeah,
so I don't know what the exact levels are. I thought the question was going to be, yes, we've measured B12 levels in the highs and lows, and they're not different. But yeah, they are not different. No, they have enough B12. I think it's just, we probably, we're pushing more into that pathway, and that's how we think propionic acids accumulate. But you know, clinically, right, if people get below, forget what is 200 picograms per mil or whatever it is, you know, that's when you start worrying about having, pernicious anemia and or, you know, neurological problems. But I'm not sure that there's good data on what the exact cutoff is, because I mean the B12 reduction in with metformin is real, but I don't know any clinician that I know that gives a lot of metformin out ever intentionally replaces with B12. I mean, maybe we're just, you know, in the middle of the country, we don't care as much, but.
Okay, there's further questions again from Sarah first. She's really interested in your findings of inheritance, showing high heritability of these phenotypes. However, a lot of other longevity studies suggest a low heritability of longevity. How do you account for this?
Well, so I'm not saying that it's the only thing, but it's one of the better things, right? And I think, and if you look through the data, those data are reproduced time and again. But that doesn't mean that, one of the things I usually put in this, this talk is that the high capacity runners, nobody likes to work with them because they're actually very aggressive animals. We actually sent them to psychiatrists, and they have a high anxiety score. And it's actually really interesting. When we did this F2 experiment, I actually, about halfway through, I went, oh, I should have thought about this. I actually asked them to score the animal before they ran them on a treadmill on how far they're going to run, because the behavior is really different between the two. So if you're a high capacity human, maybe you're a little bit more adventurous, or you have other psychological traits that put you drive faster and you get more car accidents. So there's so many things, right? Because twins don't really have a high heritability for longevity. But there's these queer things about if you break your left arm and you're right-handed, you don't really do doesn't do much of your longevity. But if you break your right arm, you actually decrease your lifespan. Now, it's just because you can't react as well and things like that. There's just a lot of different things that go into it.
Shin Wang asks, could there be a point that higher VO2 max will do harm to health? Sometimes athletes are not more healthy or younger than ordinary people.
Sometimes. Now, this is a right. What's his name? Jim Kick died of cardiomyopathy or something. So I don't know the answer to that. The exceptions may prove the rule.
Okay. Tong Shen asked, how about the behavior difference under 40% caloric restriction?
Oh, that's a great question. So I'll tell you a little story, and it'll tell you something about behavior. So when we did the caloric restriction experiment, just before we sacrificed them, we did another running capacity, and the high capacity runner, twice in a row. So we did a bunch of different nights, and we wanted to do it in the dark and stuff. But these animals are always housed two to a cage. And so we ran one to exhaustion. We put it in the cage with the other high capacity runner. It was caloric restricted, and he promptly killed his partner. And it happened twice. So we actually had to separate the animals, the high capacity animals, after they ran when they were caloric restricted, so they wouldn't kill their partner. It tells you how much difference of behavior it is.
Yeah, very aggressive. Yeah,
they're very aggressive. Now, there's a similar decrease in activity in the highs and low capacity runners. I think there's some errors in the literature about the relative activity of the highs versus lows. We think that there is not as much has been reported as if you put it in a metabolic chamber. The reason is because of their high anxiety. When you first put them in alone in a metabolic chamber, they bounce off the walls, the high capacity runners, and it looks like they have much more activity. But if you actually let them sit for about 3 days, their activity comes down to be almost identical to the low capacity runners. We did the fuel utilization becomes much closer. So the RQ, which is a little bit lower in the high capacity runners at rest, changes, but it's subtle changes.
Okay.
But they're measurable.
All right. So I don't see any further questions by the audience, but I have many questions actually.
Okay.
So let's start with some easy. On slide 10, go 2 slides up, so to say. Or,
you can see my slide?
Yeah, this is written in Koch.
Yeah.
And there is a clear jump in generation 14.
Yeah, okay.
What
yeah.
happened there?
We think two things happen. One is that when you run these guys on treadmills, it's a number. So they built, you can run 6 reps at a time, and there's a little shock plate at the bottom. We call it an encouragement strip to keep them running. And so, but the technician has to, if they start, hanging out at the bottom, you want to get the best run, so they shush them along and make sure. In here, a new technician was hired, and seriously. And the thought is that the person just wasn't aggressive enough. And so once they retrained them, it actually jumped up. Now there's one misbreeding, it looks like. Whether it affected it or not isn't quite clear.
Okay.
If you go back and read the Wren paper, Yu Wren, it's all explained in there about what happened.
Okay. So an obvious question, but nobody asked it, so, and you didn't say. You did look at number of mitochondria and also lots of places and in muscle as well as in brown fat and everywhere, right? Yeah.
Okay. So I'll go through the phenotypes. Yeah. It's just, trying to present 10 years of work in that. So the mitochondrial mass is up by mitochondrial DNA measurements. It's up about 14 to 16%. If you do it by area under electron microscope, it's similar. It's about 120%. If you just do it by protein when you get it, it's about 120%. So it's about a 20% increase in the mitochondrial mass. We don't believe it's nearly enough to cause a 10-fold increase in capacity to run.
Okay. So you also said that, the high capacity runners are somehow less efficient in utilizing energy. That points towards futile cycles in metabolism. Any idea which kind of futile cycles might be involved?
Yeah. We've been looking for that futile cycle for 10 years. We can't find it. But on the other hand, we're wondering if it's not a bunch of small things we can't measure well enough. So one of the things I showed you, at least in skeletal muscle, there's an increase in turnover, about a 50% increase in turnover of mitochondria. If you'd like, that's a futile cycle, right? You're using energy to rebuild a mitochondria. The subtle change in fuel utilization and the relative inefficiency of fatty acid oxidation versus carbohydrate oxidation over a long period of time may cause some subtle shifts in the efficiency. There's a study that has been done, is that somebody actually stuck a temperature probe in the skeletal muscle of these animals when they're running, and the high capacity runners get hotter. There's a higher temperature when they're running, again, suggesting that perhaps when they're running, the uncoupling happens more, quote, uncoupling happens more. So there may be a subtle uncoupling, there may be subtle things, but boy, I'll tell you, Oliver, we have taken lots of muscle out. We've taken a lot of mitochondria, put them into seahorses and stuff like that, give them every darn substrate we can think of, and we cannot see a consistent difference between the highs and lows.
You have not, suspiciously, you have not talked once about transporters. Now, there are a lot of people who say metabolism is all about transporters, from like Josh Rabinowitz, who talks about lactate all the time, right? Too many others. So there's no indication transporters are involved?
Well, so there are, as part of about the 1,000 genes that are upregulated these days in HCR versus LCRs, yeah, there are transporter differences. The glutamine transporter is up, OAT8 is up. I mean, there's a lot of different transporters. There seems to be a cassette of genes that are upregulated very consistently in the highs versus lows. We think we know what that actually is due to. It's probably accessibility to YY1 as a transcription factor, and it turns on this cassette of genes. And there are no doubt transporters that are associated with the phenotype, right? But when we do the genetics, we don't see, gone back and looked at, a lot of things that might be, but the genetics would suggest that it's not just one transporter or a couple transporters.
So you also said that there was a relationship of, VO2 max with longevity, obviously, and also VO2 max with obesity. But we haven't seen these kinds of relationships in lean populations or leaner populations like in Asia compared to lots of lean populations, right? Apart from, other cardiovascular events that kind of limit, of course, the longevity for for people
Yeah.
who get a stroke and so on. So if we take that out, you know, is there a relationship on VO2 max between ethnic populations? Is there?
Yeah. So you know, there's studies in Japan and in China looking at the relationship between longevity and VO2 max. It's consistent with what you see in European populations. The same changes in fuel selection, people with higher VO2 max is seen in Asians. I think I don't know if there's been one done for longevity in Indian subcontinent, but I think this is a very well-defined or well-reproduced phenotype. Not sure I'm answering your questions, but you know, most you know, the obesity relationship is quite clear that people who are obese have a lower oxidative capacity in general. Okay. It's always been questioned whether that's a cause or effect, right? And if somebody has
that there might be some metabolically healthy obese, and I understand that a lot of people challenge that position.
Yeah, so I think it's real. I think, you know, we've run, you know, 700-800 people on treadmills in our obesity clinic before and after weight loss, and we phenotyped them, and we clearly have a subpopulation that there's a relationship between your insulin sensitivity and your running capacity in the obese population. And so I have no doubt that there is variable but distinct differences in insulin sensitivity in obese populations. Now, does it protect them from everything that happens when you get obese? You know, do they have, I mean, high VO2 max decreases your risk of specific cancers. High VO2 max, you know, has all these other, you know, sort of relationships. The question is, you know, is it direct? Is it indirect? Things like that. I think it's still fascinating. I don't think we know everything. I mean, VO2 max is not the everything, but it is a good marker.
Is there any other way than putting people on a treadmill to measure VO2 max? Yeah,
so that's what we're really working hard trying to get a blood test, right? And because a combination.
It's expensive and whatnot. You have to bring people in. It's hard to get volunteers. And
Yep, absolutely. Well, you know, I can blithely say is weigh them, right? You know, what's your obesity? What's your BMI? You know, there's a good correlation between BMI and VO2 max. Now, but that's not what you really want to know because the variability is so much. You know, stuff I showed you early, you know, beginning of the talk. So, is there a blood test for it? I think a combination of metabolites might be able to do it because, you know, doing precursor product relationships and looking at those mitochondrial overloads. You know, we've been looking at peroxisomal derived like dicarboxylic fatty acids and specific hydroxy fatty acids that seem to be associated with a spillover of lipids out of the mitochondria. So we're trying to work through that, but boy, it's tough.
In the last 6 weeks, my lab has become much more interested in peroxisomes because of the long chain fatty acids, the plasmalogens. You know, so much interesting metabolism happens in peroxisomes, and you haven't mentioned it once. So looks like there's no relationship to VO2 max and or to longevity with respect to peroxisomes.
Let me just here in this.
Oh, there you go.
Slide. Okay, yeah. So we do see a subset of a consistent subset of peroxisomal enzymes upregulated in the high capacity runners. So how that relates to it, you know, boy, if you got 5 postdocs that want to come over and work on it, I'd be glad to do it.
Well, it might be. So to just give you a hint, or whatever, peroxisome seems to be preferring odd chain and branch chain CoAs and carnitines, really, compared to you know, straight chain. That also relates to the question a little bit of diet. So when we look at odd chain and branch chain, you know, fats and so on, these are usually fats that are coming from plants, that may be coming from microbes, but really, you know, different types of foods. Is there any relationship to that? Is there something you would you would tell your patients to do or not to do?
Not yet, but we're getting close. I mean, here's what we're doing. So we're I believe you're right. I think if you metabolize branched chain amino acids, or if you efficiently metabolize it, there's a signal that's being generated by either branched chain fatty acids or branched chain amino acids that tells the mitochondria that it has lots of nutrients coming in, or that it's inefficient. One of the way you can look at it either way, and that signals to the genome to make some specific subset of enzymes. The result of that is to increase fatty acid oxidation and branched chain amino acid oxidation. That's the quality of the enzyme, but it may be inefficient. And so, it's hard to bring the whole loop together yet. But we're doing the study. We're feeding high capacity and low capacity animals with propionic acid because we have some evidence that it actually in normal animals it does something. And there is some evidence that even medium chain fatty acids can cause some of these changes as well. But I don't know the composition of medium chain fatty acid diets. They're a lot plant-based, and there may be a lot more branched chain fatty acids in there as well. So, there's a lot of things to hypothesize, but I agree with you about. I think peroxisomes gives us a signal.
All right.
Of what we're metabolizing.
My very last question was about 2-phosphoglycerate. So, and your
I'm sorry,
2-PG.
2-PG. Okay.
2-phosphoglycerate.
Yeah. Okay.
Which you pasted into many different pathways in one of your last slides.
Yeah.
Yeah. So how do you even make these arguments? I think 2-PG was also in fatty acid degradation or so, and I thought like, oh, is it?
Well, so here's how I look at it. So it's if you look at what the relationship is.
So here 2-PG is an oxidative phosphorylation, and I saw it in somewhere else.
Right, and it's inversely correlated. So what does that tell you? So if you actually are not metabolizing glucose as much and you're metabolizing fatty acids more, perhaps that the lower levels of PG really reflects the change.
It's associations of a metabolite with genes that are involved in. Oh, okay.
Yes.
So then I misread it. Like, oh.
Yeah. So I apologize. This is I went over this quick, but I wanted to show it. So this is a very simple experiment looking across all these animals for the metabolite levels in their skeletal muscle, and then correlating it with the levels of the gene expression. And these are the pathways that come out, which are exactly the same pathways that we see that are differentially regulated from the highs and lows.
Then I misread this table.
Yeah, so I apologize. Yeah, I went over this pretty quick.
How did you do this pathway mapping? That doesn't make sense.
So it was just correlating the metabolite level with the gene expression levels in individual skeletal muscles.
You also promised us to talk more, to tell us about pantosinates. You did say, I'll come to that later, and I like, okay, I see it here now again.
Well, it's interesting because it always comes up, but that vitamin is intimately involved in mitochondrial metabolism as well, right? And some of the other pathways. So, it's just, maybe I said it the wrong way. I just am fascinated that you get, this kind of metabolite that's associated with these kinds of things when it seems to be derived from a vitamin. So why would that metabolism? I don't know the pathway by which it's broken down and things like that.
Well, it could be involved in CoA metabolism to some extent.
Yeah.
For example, right? But usually you'd think that nobody has vitamin deficiencies anyway, and so why would it even pop up? That's more interesting question, right?
Well, yeah. So it could be that it's the metabolism of it itself incorporating into whatever it's. Yeah.
All right. Thank you for that introduction, and thanks again for Chuck Burant spending his time with us today. Okay.
All right. Thanks, everybody. Stay safe.
Bye-bye.
Bye.